Are you all caffeined up and ready to do some bioinformatics? What I would like you to do is to have two tabs open. And so follow me on this tab here. Go into workspaces. Go into BV-BRC workshop. And so when you're beginning an analysis, this is where I'd always meet my first moment of indecision. I've sent my organism DNA off to the sequencing lab, and this is where we all are. They've sent me back the reads. And so here's the big question. Do I want to know how good a job I did or how good a job they did? So there are two services that can help you with that. Taxonomic classification is going to tell you how good a job you did in isolating that DNA and if you cleaned it up well. Fastq Utilities is going to tell you how good a job that sequencing center did with your isolates. So generally, I always start questioning other people and not myself. So I want to see how good my reads are before I accept any of the blame for what is about to befall me. Another thing you should consider too, I never really used to do this. I just always used to blow past this, assemble my reads, annotate them, do the analysis. And it was at the time when I was doing the comparative genomic analysis that I would realize, oh my, those reads were bad. This is a terrible genome. And that's generally can take you days to maybe a week to figure that out. So now it's the first thing I do. My first step is this particular service, which is the Fastq Utilities. So within the BV-BRC workshop space, which I've had you guys click to from the public workspace, click on the Fastq Utilities and click on this Fastq README for workshop.txt. I'm going to make it a little bit bigger so that everybody can see that. And then click the view here. Now, I don't expect you to read it all, but I have the SRR numbers that we're going to look at today there so that you can easily cut and paste them rather than laboriously typing SRR 1271. Okay. Second tab, here we are. We're on the home page and we're going to start with the services, which I think is what our resource is really known for and what we provide to people, which is a unique experience. Now, Malik showed you all the data, but this is your starting point for taking all that public data, picking and choosing it and intermeshing it with your private data on your path to publishing your papers. I always think of this as the path to publication, and we want you to be able to publish. We want you to be able to analyze your data well. So let's go up into tools and services, and way down over here at the very bottom is the Fastq Utilities. So let's click on that. Let's create a folder. That's the first thing we have to do on this service. So let's click on this folder icon, and you notice that what's going to pop up on yours is going to look different than on mine. This is a place where you can upload data, or you can create a new folder, or I could navigate within my workspace to a folder I wanted to put it. But I want a brand spanking new fresh folder. So I'm going to click the folder and I'm going to call it Virginia Workshop, and it's August, right? Create folder. And then what happened to it? Where did it go? Well, I can find things. Each of these are sortable. So I can click on that, and I have to click a couple times to find mine because it first sorts from the beginning of time, and then I want the most recent ones. So I highlight that, and then I click OK, and voila, there it's going to go. It's going to go right into there. Now you can subdivide and put other folders in there if you want, but there we go. Before we give it a name, let's talk about what we're going to do next. So turn your attention over to pipeline and what you are allowed to do in this service. I use this so much now when I never did before. It's become like my go-to service. So You can trim your reads. You can do paired filter. Sometimes the sequencing center, if you're doing paired reads, for some reason, sometimes the reads are uneven and you might have more on the forward strand or more on the reverse strand and they don't match. That is going to throw off the assembly service, also the variation service, sometimes the RNA-seq service. So people were, their jobs would fail, they would write to us, we'd look at it, and we would go in and manually create them to be equal for them. But now we add this service. So you can do that here. If we have a problem and you had something fail, you let me know. I say you need to use paired reads. So you can do that. Fastq is going to tell you the quality of your reads and align. I use this so much now, especially when you have mixed samples. If you know, you can map it to a reference genome, and at the end, we'll be showing you RNA-seq, a dual RNA-seq, where I have first used this to align the reads to the bacterium and also to the host genome. And then I do the RNA-seq to that. And it just makes things so much cleaner and categorized. It's awesome. And you can do any combination of these together, but I am a purist. I only like to do things one at a time. I don't want a messy job report, so I do one thing at a time. So let's go to Fastq. We're going to look at the quality, and we're going to add that in. You see, I pressed that plus icon. I could go in and I could tell it to do other things too, and we're going to do that. But right now, that's the one that we're looking for. And so let's go into that second tab, and you see this SRR number that I have highlighted here. I want you to highlight that as well, to copy it, and then go into here and put it into the output name. Well, that's what I do. I mean, you're welcome to do whatever you want, but you can see I've run over 4,000 jobs. And I found that the better I name these jobs, the easier it is for me to figure out what I was doing on that day. Because how many people are biologists in this room? Okay, how many people are computer scientists, bioinformaticians, mathematicians? There they all are. Okay. Sometimes when I'm working on these things, like I submit these things, I'm in the zone. It may be like 6 months. I'm getting my paper ready and I have to go back and report this stuff. That's why it's important to me to name it correctly and to be thinking about that at this time. This is just a word to the wise, to people in the room and Zoomers. Learn from my mistakes, name things really well so you can find them again. Okay. And so this one, I'm going to do Fastq, and I'm going to call it workshop example. And I'm not going to say anything more about it. Now down here, oh no, we can't submit the job yet. Why can't we? Because we haven't given it anything to run on. This service accepts paired reads, like Illumina short reads. It accepts single reads. It can be PacBio. It can be Nanopore. But we also accept SRA run accessions. What we were having to do before, and what I was having to do before, is download the reads from the SRA, and actually getting them from SRA is kind of hard because they don't have a friendly download for biologists. So I get the accession number from SRA. I'd go to ENA, which is the European Nucleotide Association. I download it from there because the download was more friendly to the computationally challenged, which I categorize myself as. And then I would upload it here. And each of those uploads, it was kind of dreadful because depending upon how long you leave your computer open, these files are big, guarantee a lot of failures. But now we can put it in. So do that. And then we click this little arrow to move it over into selected libraries. And so you click, and then there was this brief second of it was saying, I'm validating that number to make sure you haven't screwed up in your number. Okay, let's hit the submit button. We're ready to go because it's blue. Blue means good. Blue means we can go. So I submit it and it says it's submitting this job. Now, this is an old job that's still running for me. but down here on the jobs icon, you can see it. And sometimes it takes a while to show up in the queue. Even I get a little anxious when I don't see these numbers change right away. So I generally hang on this page until I see this. And then I may not even see it yet on the jobs page, but I learned now that when I've seen this message, everything is good to go. Now, these are read files, right? And read files are generally big. So this job is not going to take seconds or minutes. It may take hours. And when you submit a job to BV-BRC, it's not instantaneous because there is a queue. And so you enter the queue of people and you're lined up. And sometimes your jobs may take a day even to start, depending upon how heavy the usage is. So for something like assembly, I will sometimes get an email message saying, I submitted my assembly job an hour ago and it still hasn't finished. What's wrong with your system? You have to give it like 24 hours when you're dealing with reads to let it start, because it all depends. Our most popular services right now are assembly and annotation. Annotation, lightning fast. Assembly, it can take some time. If you wanted to see where the jobs were, and I don't think it's even going to show up on my jobs page yet, but let's click on that. I won't even see it registered here. It's going to take a while to move over there. Don't worry though. This is like the Julia Child cooking show. I already have a cake in the oven to show you what it looks like. So let's go into workspaces and let's click on BV-BRC workshop. Let's go back to Fastq Utilities and let's go into Fastq example with good reads. So I'm going to click on that. And so you see that there's this title here with this weird icon here, this checkered flag. That's supposed to be reminiscent of NASCAR or Formula One, meaning the race, you cross the finish line. This is a completed job when you see this. So let's click on that and we can actually double click on it and it'll open up the job page for this. Now we ran it through Fastq Utilities, and although it was just an SRR number, right? Now I can see that it was a paired read because it's given me two different reports on the read files, and it's also giving me some analysis on this. So if I highlighted this text row and click the view icon, it's going to tell me everything I wanted to know about those reads. Then, so it's bringing all the data from SRA to tell me what I need to know. And that's forever wed with this in your jobs page. Any medical entomologists in the room? Oh, I am one. And so today you are going to have a wonderful experience of all the insect endosymbionts that I love. You too. I'm sure everyone will throw off AMR or pathogens destroying the world. You're all going to walk away from that and turn your attention to this when you figure out how cool these guys are. Pediculus humanus is the human body louse. And this is a metagenome from that louse. And that's why I chose it. And I also chose it. We're going to work on those a lot today because their genomes are small and they assemble really quickly. So everything will work a lot faster with those. And they are the coolest organisms on the planet. So that's just the reason for that. Up here, this is a breadcrumb. So we can go back up to it. Let's click on this, the next one up, and it'll take me back where I was. And then we also provide information about this read file and looking at the quality of it. So this will be the forward strand. So I highlight that, and then I click on this magical view icon. You notice when you click on things, it populates this vertical green bar with possible downstream actions of things that you can do. So let's click on the view icon. And then this is the FastQC report. And it tells me what I need to know about this. This was Sanger Illumina, how big it is, the sequence length, and the percent GC. And so this is, and I'm going to give it more room here so I can show you guys. I'm going to move this to the side. This is actually the main place I stop here. Because when I see that everything is in the green, pretty much, this guy at the very end dips down into the poor quality. Here, let me shrink it up just a bit so you can see more of it. So these look like great reads. Along the side, it tells you areas that the FastQ pipeline might have found some problems with, just to notify you with, but nothing serious here. The per tile sequence quality is giving you an indication. It's looking at what your sequencer did, and there might be a particular tile within the sequencer that was bad. If that's true, you'll see sometimes a big red block of things that you can go back. You could download this sequence report, send it back to your sequencing facility and say, I paid you this money for these reads, and it says that something's bad. You guys have to rerun it and give me something good this time. The per sequence quality scores, those look good. The per sequence base sequence content, those look pretty good. And you're going to think, how do I know those look good? I'm going to show you what a bad one looks like in just a second. This looks good. You also want to see, you always want to see the GC content mirroring the theoretical distribution with your particular sample. Did it find any N's? It's looking through your reads and finding an N. Who knows what an N is in a read? Say it and earn your first chocolate of the day.
N would be an anonymous read where you're not seeing.
Right. The sequencer, when you see an N in there, it means that the sequencer couldn't get the signal. It couldn't figure out if it was an A, a C, a T, or a G. So it'll put an N in. You sometimes find these things, and people to actually close a genome will sometimes have to resort to PCR to get past difficult regions to sequence those to try to find things. Does anybody know the difference between WGS and a closed genome? Yes.
Complete genome with no gaps.
Right. The complete genome has no gaps, but also technically, from what GenBank says they require, all the N's should be resolved in that. WGS is whole genome shotgun, which is just, it's all in pieces and contigs. So when there's no gaps, when it's complete, and those are supposed to be the higher quality genomes for your analysis when you're comparing it to things, that's meaning that the number of contigs match the number of chromosomes in the organisms. And if it's truly complete, no ends. All right. Sequence length distribution. So you're seeing pretty much what this is. And did it find any duplication levels? It looked like a little bit, but I'm not too worried about that. And overrepresented sequences, this would be where it would tell you, oops, you've got some trimming to do, that there are some problems here. And then you could use the trim service. So let's use the breadcrumb and go back to Fastq Utilities here, right there. And then let's click on Fastq example with bad reads. And then click on this guy, double click on it. And so this was from Frankfurt, Germany, a wastewater sample that I got from the Sequence Read Archive. And then look at this. Nobody's in the green. And look at all these warnings here along the side. This looks terrible. So it doesn't like the per quality sequence scores. Remember how crisp and clear that looked on the Goodreads? This doesn't look very good. Oh, and look at this. The red is this sample compared to what it should look like. The ends, at least there are not too many ends. The sequence length distribution, this is all over the place. Remember how nice that was before with that 101 in the center, and it's a very pleasant distribution. This looks terrible. Duplication. And then it's telling you there are a lot of overrepresented sequences. And you know what? I have tried trimming this. I've tried a lot of things with this thing. This is just not a great sample except for one thing. If you work with SARS coronavirus, this is a really great sample. So you could align the data to the Wuhan reference genome. And if you wanted to pull it out of that, you could do that. So with this particular sample, I also ran it in taxonomic classification, and I could see it had a strong SARS component. So you could pull that out. Although we're not going to work on that for this next section, right now we're going to show you how to do an alignment and align your reads to it. And it's not like a genome alignment. It's just going to be mapping those reads to the contigs and throwing away everything else. Well, doesn't throw away everything else, but we'll show you that. So let's go back to tools and services, and let's click on Fastq Utilities there at the bottom again. We know our output folder. Now you don't have to navigate to it. If you have just created it, you can click this down arrow and it'll be at the top. You've got a hint of what my workspace looks like and how much stuff is there. If I wanted to put it someplace else, I would try to remember. And so I would just start typing in there, and it's going to find everything for me in my workspace that sort of matches that. So there are 3 ways to get there. You navigate, you use the drop down, or you commence typing and you've got it. Now let's go over to your second tab that has the README open of it. Oh, I should have had you submit the poor example. That's your homework if you so desire, so you can try that. So this is the alignment example. So let's grab this and put that, I want to put it in the output name. And before we choose the action, let's just go down, paste it there, and then move it over. So it went pretty quickly. Now we need a pipeline, and I will have a new tab, and I'm going to put in sequence read archive here. You don't have to do this. I just want to show you what you can do with my examples if you don't know what they are. How many people have ever used the sequence read archive? It's awesome, isn't it? The amount of data that's in there, all the stuff that you can compare things to, it's amazing. So if I search for that, let me blow it up so you can read it. This is the whole genome sequence of Glossina morsitans submorsitans. Anybody know what that is? It's the tsetse fly. So we have another endosymbiont. And let me tell you that the tsetse fly, let me just wax on a minute for you while I try to convert you into insect endosymbionts. In my opinion, this is the most important organism in Africa. Now, why is that? Does anybody know what the tsetse fly transmits? African sleeping sickness. All the large land mammals in Africa on the plains of the Serengeti, they all have sleeping sickness, but they have co-evolved, so it's no problem for them. And for hundreds of years, Europeans have tried to put cattle to take advantage of all that grassland. They die immediately once they get bitten by this guy and infected, because they have not co-evolved with the organism. So for those of us who like biological diversity, especially in Africa, this should be your hero. So we should all get a little tag that says, I heart tsetses. Okay, enough on that public service. Align to endosymbiont. Okay, so that's what I'm going to call this thing. And then we're going to go into the pipeline and click the align button. And then we move it down there. So it's in the alignment thing. But notice I did the same thing as before. The submit button isn't blue, and it's asking me for a target genome. Well, I already know what's in here because actually I first ran this on taxonomic classification because it wasn't my data. So I didn't want to blame anybody else. It was SRA's data. So I'm okay with blaming them. So let's go back to this other tab that we have open. And there's a genome ID number here. So just go ahead and copy that. But I want to show you what you can also do. Well, I'm going to map this to is to a particular endosymbiont that's known in this organism and that I know is there. And I want to open this thing up because I've done this so many times. I want to turn off my genomes. You don't have to do that, but I'm going to turn off mine so you don't have to see how many times I've worked with this particular organism. And the name of this organism is Wigglesworthia. And I want to see if it's going to pull it up. Good. You could either put the genome ID in there or see how fast and smart this was to be able to find this thing. So I could put that in there. Now we don't have the tsetse fly genome. We can't use that as a reference genome, but I can use this. So now I've got that, but I could have just as easily, if I take that out, I could have cut and paste that genome ID in there, which is a unique identifier. Look at what it gave me. And you might say, if it's a unique identifier, why did it give me 3 of those? Well, you can see that the numbers are also contained in the other ones that are in there. This 36868.4 is also part of those influenza ones. So then I can't do everything for you. You need to be able to click on Wigglesworthia yourself, but I know you can do it. I can see that you all have that ability. So click on that, and then we can submit that job. And that job has been submitted, and let's wait until I see it. Oh, good. And then it's been successfully submitted. And normally I'd be very concerned when I wouldn't see that even up. Look, it just popped up. I was just about to complain to the developers in the room about this, but before I could even get it out of my mouth, it popped up. So the jobs are running. So this time you can always click on the job monitors, but I could go up to my workspace, my jobs, and see it there. Oh, look, the first Fastq QC finished running. And now it's in the queue. Question. The question was, you wanted to confirm that the target genome that we used, that was the reference genome that we're mapping to. And I just want to make sure to distinguish between references and the GenBank references that Malik talked about earlier. In this case, it's whatever reference you choose it to be. And it could be a genome within the public sphere, or it can be your private data too. If you've assembled this and annotated this in our system, that can be your reference as well, which is very helpful, like when you're doing RNA-seq of your own novel organism or whatever it is, you can use that here and map those reads there. Okay, we're back to our K, Julia Childs. Let's go into workspaces. Let's go into BV-BRC workshop. Find Fastq Utilities, click on that, and then click on alignment. Let's click on the Wigglesworthia. Click on the folder that said reads that map to Glossina, and then align to Wigglesworthia. And I said morsitans, which is, I must have been eating too much chocolate and not thinking about it. And Wigglesworthia, if anybody is interested, this genus is named for the famous insect physiologist Vincent Wigglesworth. So if you want to become immortal, sometimes it'll occur as a bacterium. So when we ran this job, we got a number of things. So we got BAM and BAM.bai files. Does anybody know what you can use a BAM file for? Anybody who's not part of the group? We can look at this on a genome browser and see the extent to which it has mapped to this genome. And we'll do it in a minute. We'll go up to this browser and look at that. So it'll give you a sense of how deep the sequencing reads are for this genome. Then we have FQGZ. We have 2 of those. They're not exactly together, but one has this 2 in here and one doesn't. Who knows what FQ.gz means? It's a zipped file, a Fastq reads file that's been zipped. And we also have unmapped. So what the service did was we gave it a boatload of reads and said, separate the wheat from the chaff. For us, the wheat, if you don't get the reference, sorry, or I'm not sorry, but I will say a lot of weird things that people will be thinking, wheat from chaff, what's that all about? And just let it flow over you and say, it's just her. I'm just going to listen. We asked it to map to Wigglesworthia. So those are the aligned reads. The unmapped reads are all the other stuff. So there might be other things in there. Question.
In the case of genomes that are like diverse, does this have the option of mapping to multiple reads?
You mean multiple genomes at a time?
Yeah.
Yeah. Well, what I could do and what I have done with this service is I've run taxonomic classification and seen, oh, it looks like it's these things. So I run the same thing to species 1, species 2, species 3. If this was from a human being, I could map it to, and in fact, the RNA-seq example that we will have at the end of day 2, that was a previous thing where we took those reads from Orientia tsutsugamushi, another wonderful little bacterium that you're all going to love. But this one, it's not an endosymbiont, but it is the causative agent of scrub typhus. So I mapped it, ran the same reads, mapped it once to Orientia, mapped it to human, but I've taken the same example and mapped it to Rickettsia, to Coxiella, and I believe I had Bartonella in there too. So 3 different ones from the same set, and then I had those reads aligned. But we have another service we're going to talk to you about today called metagenomic binning, where you can just submit it to that and it's going to take them all out. And if it can pull up a complete genome for you, it will do it. So that's later today. Does that answer your question?
Great, thank you.
Okay. Yes, question. The question was, what was the difference between this one, which is aligned without this 2 here, and this one? So it's the forward and the reverse strand. So it was paired reads, and this 2 means that's the second strand. And this one, it would be less confusing if it had a 1 in there. So you'd be able to see that like it does here. But, you know, I guess it's on our list of things to do. So we haven't gotten there yet. We've been the other stuff. There's also that meta text of things that you want to see. So first, oh yeah, and then there's this, which is the report on the alignment. And it's going to tell you how good that alignment was to that genome. So you click on that and you click the view icon. Now, some people in the room think this is a really dumb thing, but I really like it. It's so cute. And you look at it and it says unmapped. And so if I were to just look at that, I would be like, oh no, this totally failed. Look at that. So few reads are in this thing. This is awful. But if I go down under the high map quality, even though it's 0.1% of these reads, we have 5,000 reads that mapped. And then these are not too bad. But even though it's small numbers, let me tell you that this is enough to get a pretty good genome out of this. And when you think about all this unmapped reads, what do you think that is? Remember what this sample was? It was a squished tsetse fly. So all of those unmapped reads are probably tsetse fly, which we can't actually do anything with in the BV-BRC system, but just to know. And okay, and this report goes on and on and on and on. I never look at all this stuff. It goes on forever. I just look at it for that cute little circle that pops up to show you that. And then trust me, because I've done a lot of trying to game this system, because my goal is always get the best genome I can for my analysis. So for this particular one, which is an endosymbiont that's very small, these are enough reads to get you a pretty good genome. Now let's go back, and I may have to go all the way back up to this one, reads that map to Glossina, and click on this again. So the reads here, those are already integrated within the BV-BRC workspace. So you can use these reads in downstream analyses. Just like that, you can either navigate to this place, or it's going to remember what they are from your most recent jobs, and you'll be able to see them in that drop down box right away. Or you could click on the name and search for it. And I'll just show you that really quickly here, even though we're going to step away from this one. But let's say if I was in the assembly one and I wanted to use these, and see it's automatically looking for the reads that are available to me. And now you can see the dirty secret is I've run this a lot of times. So I have a lot of that in there. So you'll be able to run those things yourself when your job completes. So another thing we have up here is the genome browser. So let's click on that. And Malik stepped you through this, and it's showing you the ref seek and the BV-BRC annotation. And you'll notice that, I want to bring the box over. You'll notice that it's got those aligned reads there, the BAM and the .bai file. The BAM file are actually the reads. The .bai file is important for this browser to be able to see them. Now, if I click on that right now, it's going to be a mess because it's too much data. So I need to zero in on it and just click this several times. This one might be good. And then click on that, and it's loading it. Come on. There we go. Let me remove this so we can see it better. So these are the reads, and this is showing you how intense to your reference genome, how many reads you have. And so you could scroll down, you know, if you wanted to see other places, and it's loading the reads. There are fun things if you're interested that you can do, like hide reads aligned to the forward strand. And then you see all of that. And that always surprises me because I always would think the forward strand should be blue and not red. I don't know why I think that, but I did. But that's the kind of stuff that you can do. I want to go back to this job again and Fastq Utilities. And I think I have one more example of a good alignment example that I have since thought was not a good example. And you can see the breadcrumb up here tells you where you are. So let's click on that one. And then this one, I want to see the view here. Look at this. And then, oh, you look at that and you think, oh man, this is beautiful. This is just absolutely stupendous. But here's a word to the wise. You always have to do all the other trusting too. So let's click back here, back on good alignment example, and re-click on this. And I'm just doing this so you can see the browser. And so this was an example for Borrelia burgdorferi. Who knows that genome? Causative agent of Lyme disease. Oh, well, this is pretty sad.
That's how did you go to this page from the previous page?
All right, the question was, where are you? I'm totally lost. How did you get to that browser page? So let me go back to the workspaces. BV-BRC workshop, Fastq Utilities, alignment, and then the good alignment example, and then double click on the checkered flag. Up at the top, there's a view icon and a browser icon. The view icon is going to take you to the HTML report. The browser icon will take you to the browser. The pie chart is here in the view. So if I click on that, there's the pie chart. And now I'm going to try to hit the back button. Okay. And the reason I did that is because if I navigate up in the breadcrumb, I can't see these. I have to do it through going through the jobs thing. And I don't know why that is, but that's a fact that I've learned to live with and not complain about. And so when I want to see the browser, then I click there. Are we good? But I'm going to go back here where I've zoomed in on it. And then I click on the aligned sequences and I wait for them to load. And look, it did load. That's it. That's it, maybe for this. But look at this. This genome is in multiple contigs. So you would have to go shopping for that. But if this was the area of the genome that you were interested in, which is the Borrelia plasmid replication region protein 4, it had 2 reads that mapped to that. But if we went shopping and went to something different, and who knows, so you click here, and then these are all the contigs associated with this particular genome. So it is not a closed genome because Borrelia is in a single, well, actually, Borrelia has a ton of plasmids. So it could be one of the plasmids. And if I were to click on that, but you can see that even though, remember that pie diagram and everybody got excited? I used to get excited too, until I clicked on the browser. And then I thought, oh, maybe this stinks. But I wouldn't go that far. But that's why all the knowledge is there at your fingertips for this. Look at that. So sad how few reads there are to some of these things.